The following audio recording has been designed for learning purposes only. Do not share, distribute, or provide any type of copies, or this audio lesson will not be made available to you. Good luck with today's lesson.
Just want to make sure I didn't miss anybody. Is there anyone who's new who does not have a syllabus yet? Okay, today's the last day I have to ask that because yesterday was the last day to add, so whoever's here is here now. I am going to pass around the attendance sheet. This will be the one we use for the rest of the term. So like last week, just initial under today's date. That's how I'll keep track of who is here. Before we get started, I'll always try to give you guys an overview of like what's coming up today, what's going on this week, all of that, so that you kind of remember, not only from the schedule and the calendar on Canvas, but just because I'll remind you too, what's coming up. So today on the agenda, we are going to relatively quickly finish chapter 1 and then roll right into chapter 2, start talking about some chemistry stuff. When we get to lab, remember you have your pre-quiz today on labs 2 and 3. Don't freak out if you're new. I'll explain more about that during break. We'll take a break in the middle, so I'll catch you up on that. Labs 2 and 3, so if you forgot about it, you got a 10-minute break. You've got a break before lab, so just look over those labs if you had forgotten to do so this weekend. The lab that we're going to do today, I think I'm going to start us off with lab number 2, and we'll do 3 on Wednesday. Thinking ahead, like planning ahead for next week. Remember, next week, Monday, we don't have class, MLK Day, so you get an extra day off, nice long weekend. If you didn't know, the college has some really cool volunteer opportunities, so if you're interested in taking part, there are some on Saturday and some on Monday. You can check the website if you're not sure and you're interested. Any questions on what's coming up today? Or any questions from anything we did last week, either in lecture or in lab? Okay, I'm going to close that door so we don't get the noise from the hallway, and then we'll jump back in. So I think this is the slide that we left off on, going over the scientific method. Most of you indicated that you have learned this somewhere before. So hopefully we're just gonna kind of refresh everyone's memory, and we won't have to spend too long on this. What I believe we did already was kind of went over the basic steps involved in most of the process, right? You make some kind of observation, you notice something you're curious about, do a little research, what do people already know about this? You then try to come up with an idea to add something new to the conversation. Like, what do you think is the possible explanation for what's going on? Remember, the most important thing about your hypothesis is that it needs to be testable, because that's what your experiments are designed to do. Test your hypothesis, which means every way that you can, every way you can try to imagine, you're going to try to prove your best guess wrong in any way possible. Your conclusion, based on how those experiments go, remember, is always going to be one of two things. Either your hypothesis was correct, your data support it, or your hypothesis was wrong, your data do not support it. Either way, we talked about, I think, the fact that you're probably going to come back up here and continue this cycle. This little circle going on right here, that's the real work in science. It's a constant refinement, a constant asking the next question, see what more we can learn. Questions about that really brief recap of what we went over last week. Okay, I think where we had left off was we didn't talk about theory yet, or did we? Maybe we did in here. Did we talk about theories? Does anyone remember? We'll just talk about them again, just in case. All right, thank you. So theory, remember, I usually always start here by saying a theory is a word that's used differently in science versus not. If we're just like layman's terms talking to our friend and we say we have a theory about something, it sounds like it's just a random guess, like, oh, I'm not really sure, but here's one crazy idea. But in science, theory means something very different than that. Something is only a theory in science if many experiments have been done over the course, usually of a long period of time, by many different scientists in different fields, done in different ways, etc. And all of those observations kind of all point in the same direction. That no matter how hard everybody has been trying, nobody has been able to poke holes in this idea. And instead, contrary to that, all of the evidence has been mounting and mounting and mounting that all leads us to believe, given everything we know, this is the best explanation for what's going on, right? Gravity is a theory. Cell theory, the fact that all living things are made of cells, is a theory. Evolution, so when I always mention here, evolution is a theory as well, right? So the point I always make, I may have said this last week even, is that I don't know anyone who doesn't believe in gravity or believe in cells, but it's not hard to find people who don't believe in evolution. Yet it is a theory, so there's the same kind of volume or same body of evidence supporting that theory as there are the others that we more willingly accept perhaps. So we'll explore some of those ideas, maybe why that's the case when we talk about evolution later. Any questions on what a theory is? Okay, so the slides that follow, I've got kind of a slide on every one of those steps in the scientific method. And I think probably what I said at the end of class last week is that instead of walking you through the sort of sciencey example that you'll find in the notes, what I want to do is offer you a non-science example of how we can explore some of the important vocabulary associated with the scientific method. So the silly example I want us to think about, which I think can be illustrative, is baking a cake, right? So here's like the whole backstory. Let's say every year on your birthday, you bake the same cake. It's like your grandma's recipe or whatever, but you really only bake it like that one time every year. It's like a very special cake, right? And let's say your birthday is coming up, but maybe your sister just had her birthday, and she calls you and says, you know what, I like messed up when I was baking grandma's cake, and I accidentally added 2 eggs to the recipe instead of the 1 that it calls for, and I gotta tell you, I think it tasted even better than before, right? So there's maybe the observation that we start with. It's based on, you know, my sister's telling me the story that 2 eggs is better than 1. Now, I'm all about delicious cake, so if there's something I can do to improve the cake I usually bake, I want to see what's going on with that, right? So what do? Thank you. I come up with a hypothesis. How can we test this idea? How can we figure out what's going on here? Given the setup I've kind of explained already, I'm hoping our hypothesis would be fairly straightforward in this case, right? Baking my birthday cake with 2 eggs instead of 1 will make it taste better, right? Again, if your hypothesis is well written, it sort of designs your experiment for you. And it also kind of makes a prediction about what you think will happen, right? My hypothesis did not say the cakes will taste different. It said very clearly which one I expect to be better. I expect 2 to be better than 1. I didn't just say they'll be different, right? That's a well-written hypothesis, again, sort of making a prediction about what should happen. And as I've already stressed, the single most important thing about any hypothesis is that it must be testable. Again, that's what your experiments are designed to do, to try to really test this hypothesis. So, in this crazy cake hypothesis world, how would we actually test this hypothesis? What would we do? What would you do if you wanted to know if the cake tasted better? You bake the cake, right? But you have to always perform your experiments in a very carefully controlled manner. Honestly, these are the 4 vocab terms I really want to go over with you guys. This is kind of the heart of the matter in making sure you design experiments that will ensure that your results are trustworthy, right? I think where I want to start is actually these 2, control group versus experimental group. If we're going to bake that birthday cake with 2 eggs in it, are you going to know for sure if it tasted better than a cake you made a year ago? No, probably not, right? I can barely remember what I had for dinner 3 days ago, let alone what a cake I had last year tasted like, down to like the exact flavor and crumb texture and whatever, right? So if I want to really know if that 2-egg cake is better, what do I have to do? Make the 1-egg cake to compare it to, right? This is the importance of a control group. The control group is essentially something you're going to use for comparison, where you haven't changed anything. This is like your regular set of circumstances. That's our 1-egg cake. And then your experimental group is where you're kind of switching things up a little bit. You're trying to see if something you're doing is having an effect. So that would be our 2-egg cake. Now, the fact that we have 2 different groups, in this example, 2 different cakes, means that we must be changing something between those 2 groups. And thinking about what we're changing, that's the importance of these other vocab terms. Experimental variable singular, just one of them, versus control variables. And I tried to kind of highlight that one as plural by stressing it when I said it, but also kind of putting it in italics and bold there at the end. So what are those terms all about? Well, if we really want to know if these two cakes taste differently, if we really want to know that it's due to the eggs, the number of eggs, that can be the only thing we change between the two cakes, right? You have only one experimental variable because that is the one thing you're changing between your two groups. Everything else that you can think of, you must purposefully keep the same between the two cakes, right? If I change multiple factors between the two cakes and then the cakes taste different, how do I know why they taste different? It could be due to any one of the things that I changed. But if I only change one thing and the cakes taste different, I know exactly what, is to blame for the difference or what to credit the difference to. So if we were actually going to be baking these cakes, what are some examples of things that we would have to make sure we kept the same between both groups, the 1 egg cake and the 2 egg cake? Batter, the other ingredients, perfect, right? Let's get like real specific here because this is what you'd have to do. Let's think about flour, right? What do we have to keep the same? The same amount, perfect. I can't use 2 cups in one and 1.5 cups in the other. The brand, perfect. I'm gonna go a little bit dig a little deeper there. Even the type of flour, right? I can't use whole wheat flour in one cake and like super fancy baking flour, whatever, in the other cake. The brand, I would say even down to the bag of flour that you're using, if you can use the same one, right? I shouldn't use a bag of 2-year-old stale flour in one cake and a brand new, freshly opened flour in the other. Like every factor you can consider, not just the flour, but every other ingredient too, right? Can't use 1 cup of brown sugar in one cake and 1.5 cups of white sugar in the other. Can't use butter in one cake and oil in the other. Every other ingredient, every aspect of every other ingredient must be kept the same. How about other than ingredients, what else would we have to control for?
You'd have to have the same temperature to make
Perfect. Yep, you can't bake one cake at 450 and one at 375. Can't bake one cake for an hour and one for 45 minutes. Can't bake one in a glass square pan and one in a metal circle pan, right? I'm talking every aspect you can consider must be controlled. Remember, a control variable, here's the definition if you didn't jot it down. Control variables are applied in the same exact way to both of your groups. They're things that you are keeping constant between your control group and your experimental group. If you make sure it's applied the same way to both groups, you know it cannot impact if there's a difference in outcome because it was the same across the board. You very purposefully design your experiments such that there is only one thing, the experimental variable, that you're changing between the two groups. That way, again, in this example, if your cake tastes different, you know exactly why. Because the only difference is that you added 1 egg to the one and 2 eggs to the other. These 4 are always the definitions associated with the scientific method that I focus on, almost exclusively when we go over this, because I think the basic steps people are pretty familiar with. Yeah, make an educated guess, do some experiments, whatever. This is really how you ensure that you're performing your experiments in a carefully controlled manner to guarantee that your results are meaningful and trustworthy. Any questions on those 4 important definitions? Yeah.
Can you repeat that control
variables? Yeah, they are, situations or conditions, whatever it might be, that are applied in the same exact way to both the control group and the experimental group. Okay, so it's basically things you're keeping the same between your different groups. Good. Other questions? Okay, cool. So we bake our cakes. We bake our cakes, keeping all our variables how they need to be, and we come to some kind of conclusion. Now remember, your conclusion can always be stated one of 2 ways. Either the data support the hypothesis, or they do not, right? So let's say that 2 egg cake was better than the normal way to bake the cake. In that case, our hypothesis was that it would be better. So if it was better, I could just say the data support the hypothesis. But I always say, like, if someone came into class just a little bit late and all I was saying was the data support the hypothesis, do they have any idea what kind of experiment we were doing? No, right? This is why you always want to state your conclusion as specifically as possible. Instead of simply saying the data support the hypothesis, if I were to say, baking my birthday cake with 2 eggs made it taste better than baking it with 1 egg, you've got kind of everything in that one statement. You know what we were doing, baking cakes. You know what we were changing, the number of eggs. You know the outcome, 2 is better than 1, right? In one carefully crafted conclusion, you get all you need to know. Even if you miss the whole setup of the whole deal before that, that one statement captures everything that we were doing. In baking and in science, all details must be recorded and reported. Why is that important? Go ahead.
Kind of going back to your statement earlier.
Yeah.
If someone like walks in or
Mhm.
whatever else, and they don't know, like, they'll hear the result, but they're like, well, how did we get?
Yeah, exactly.
Yeah.
So I always like to start with the baking side of this, right?
Yeah.
Because there we're a little bit more used to the idea of a recipe and
Yeah.
needing to know exactly what to do and what to add, right? If I told you all, again, let's talk extra credit. If I were to say, go home and bake me my cake and bring it back Wednesday for 50 points of extra credit, could you do that if I didn't give you the recipe? Of course not. There are like a million cake recipes online. You wouldn't know which one was the right one unless I told you. Going away from baking into science, we have to share results in science. Why? Other scientists work off of each other's previous findings, right? If I had spent 20 years working on a certain topic and never shared my results with anybody else, did they really happen? Like, are they really meaningful? If I'm the only scientist that can ever get the results that I get, how trustworthy are those? I don't know, right? Instead, what we need to do is share exactly how we do our work so that another scientist somewhere else could duplicate it. If no one else can follow your steps and get the same results that you got, that would call your results into question. Right? So if you've ever read like a scientific journal article, not like one written by a journalist for like general mass consumption, but like an actual paper written by the scientist who did the experiments, they're very technical and difficult to get through because they have to include all of the details about everything, right? Like what incubation, what temperature, what concentration of that solution, blah, 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 a lot of detail. They have to share that so that other scientists can try to recreate their work. This also, of course, saves people a bunch of time. Am I gonna waste a year trying to carefully design an experiment if I know it's already been done? No, I would look at what has been done, maybe try to recreate it myself to see, yes, that does work, before I can then move on to something different. You always have to record everything in absolute detail. Every minuscule detail you have to record and share so that other scientists can hope to recreate your work. Any questions about that? All right, I'm going to skip this slide on theories because we already talked about it. I already made the point I want to make here, so don't cross this out. Keep it in if you have slides. I feel like we already said what we need to say about it. And here are a handful of slides that I put in here in the lecture notes that are the more like sciencey example of the scientific method. I don't feel the need to spend 10 or 15 minutes walking us through it because I was going to use it to illustrate the same exact points I did with like the cake baking example. I like doing that example because it's maybe more of a relatable way to think about why these different factors like control group versus experimental group and control variables, plural, versus the one experimental variable. I feel like the cake one is a good way to kind of make it more relatable. If you want another example, all of the details are here in the slides. You can go over it. You can ask me if you have questions about it. I'm not going to ask you anything specific to this example, which has to do with like adding fertilizer to see how much crops you can grow. I'm not going to ask you anything about this specific example. It's much more important that you understand the process and how to properly apply the steps. So I'm going to just kind of fly through these slides just so you can confirm, hey, if you want to see another example, walking you through them is all here with this other example. And you can always let me know if you have questions. And that will essentially do it for us in chapter 1. So this was, we took a while on this chapter because I used it to also kind of set you up for the whole semester, give you an idea of all the different topics we'd be covering. Remember, we started this chapter initially a week ago by acknowledging that there are millions of different species on the planet. And even as different as all these species are, what we did was we built a list of common features, things that all species have in common, this list of requirements or characteristics for life, right? For example, I'm not going to list them all, but just some of the big ones. All living things have that organizational system we looked at, atoms, molecules, cells, etc. All living things have to get materials and energy. All living things have to reproduce, maintain homeostasis, etc. After that, we came back to acknowledging, wow, there are a lot of different living things. How do scientists keep track of all of them? Remember, taxonomy is this field where we essentially group species based on similar features. We put them into a domain if they can do this, and then a certain kingdom if they can do this and that and whatever. It's a way that we group like things together. We're going to be talking a lot more about taxonomy in lab this week, in actually both of the labs that we're going to be doing. So I'll reference that again later today. A little bit about evolution was kind of sprinkled in various parts of the chapter. We talked about natural selection, survival of the fittest, that whole idea. And of course, we rounded it out by looking at the scientific method. Does anyone have any questions on chapter 1 before we move on to chapter 2? Yeah, go ahead.
This kind of goes back to the lab work.
Yeah.
Will we be going through like the homework assignments, like if they were right or wrong? Or
Oh, like the calculations you did in lab last week? Yes. So I forgot to address that in lab last week. I think someone may have asked me at the end of class, but a bunch of people were already gone. So you're not handing your lab sheets in for points. So lab 1's a little bit weird in that it's just a whole bunch of calculations and not any like summary questions. So I am always more than happy to look at your data sheets and check that for you. Like I have a little answer key for all the calculations. I can quickly check yours. I'd be happy to do that for you either today or whenever. A lot of the other labs that we'll be doing do have some questions associated with them, and I'll always try to make time for us to go over them together as a class at the end, just to make sure everyone sort of got what they needed to from the lab. Cool, good. All right, other questions? All right, so we're going to go ahead and move on to chapter 2, which is our first foray into the wonderful world of chemistry. Yay, exciting stuff. Okay, so chemistry is not a prereq for the class, right? Just want to make sure we're all aware of that. I like to ask, just to kind of get a feel for it, who in here has taken chemistry somewhere before? High school, here, somewhere, anywhere? Okay, that's a lot of hands that went up. I always just like to try to get a feel for the room, like read the crowd a little bit, because if you've ever had chemistry somewhere before, I think that you'll find a lot of chapter 2, maybe all of it, is review, to kind of maybe refresh your memory, bring you back up to speed, whatever the case may be. However, if your hand didn't go up, or if you've taken chemistry, like 5 years ago or more, and you just totally forgot everything about it, don't worry. We're going to go over all of the basics together. You just go into this chapter knowing, all right, this might be one where I have to spend a little bit more time kind of going over it than someone who just had chemistry last semester or last year. Remember, this is what I said last week, like always think about, where am I at? What do I have to do to get up to speed? If you know you're pretty solid in chemistry, maybe you don't have to spend quite so much time on this chapter. If you know I have no idea what the hell she's talking about right now, might be one where you need a little bit more time on this. What are we going to go over? Again, I think the basics. We're going to start off by talking about atoms, the fact that they make up everything on the planet, living stuff and non-living stuff. And we're going to go over the parts that make up atoms, protons, neutrons, electrons, sort of big properties about those so-called subatomic particles and how they allow us to figure out why atoms behave the way that they do. Thinking about an atom's behavior will lead us into sort of appreciating how and why molecules can be formed through different kinds of bonds. It all comes down to individual atoms and how they're behaving. So we're going to focus today on a couple major kinds of bonds that hold molecules together. Ionic bonds and covalent bonds, there's a big difference between them, so we'll make sure we kind of get that figured out. And there's a whole section of this chapter dedicated to the chemistry of water. I have cherry-picked just a couple of properties of water that I want us to focus on. The rest will be stuff that we'll probably just skip over. And of course, as I always will try to do, I'll let you know what of that you need to know about and what of that we're not going to worry about. So we're going to start in the beginning with a definition for matter. Pretty straightforward definition, I think. Matter is anything that has mass or weighs something and occupies space. So if it takes up space and if it has mass, it is matter. Right, so I always start this with just a simple question. What in this room is matter? Everything. That's my best answer, probably. Everything, right? Tables and chairs, which are not alive, absolutely. Us, which are alive, absolutely. Everything in here, even the air, which people sometimes forget about because you can't really see it. It's taking up every inch of available space that's not filled by something else. And even though it's lighter than us, it does weigh something. So everything in here is matter. And all of this matter, whether it's living or not, is made up of the same 92 naturally occurring elements. We don't have a periodic table in the room to look at, but if you've seen one before, if you've taken chemistry, you might be aware of the fact that there are more than 92 elements. So in case you are aware of that, it's not something I'll ever ask you, but in case you are aware of that, the reason there's a number discrepancy is that some of those elements have been man-made, 92 are naturally occurring. Elements are essentially a type of atom. It's a substance that has a certain set of properties that it always carries with it. It behaves in a predictable manner. Of those 92, just six of them, make up 95% of our body weight. In case you don't know what these abbreviations are, I'm not going to ask you what these are, I don't think, at any point, but just so we're all on the same page: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The only reason, honestly, the only reason I'm even mentioning this, why I put it on the slide, is because after we do chapter 3 on Wednesday, I'll remind you of this list of 6, and at that point you'll know why these 6 are the 6. Now obviously, that's not 100%. So there are other things like iron and calcium and stuff like that that's clearly important to us as well, but just bulk weight-wise, we're mostly this stuff. Okay, so those are going to clearly be important atoms that we're going to be seeing a lot of, not only today, but also in chapter 3. Remember, chapter 3 that we'll do Wednesday is about some really big, really important molecules like proteins and carbohydrates and DNA. So we'll see a lot of these atoms in those big molecules. What we're going to do is focus right in on atoms. An atom is essentially the smallest chunk of an element that has the properties associated with that element. For example, if I had just a box of carbon up here, whatever that might look like, I've just got a box full of carbon. An atom is the littlest piece I could reach in and hold up, and you could still say, oh, that's carbon. Absolutely, I can identify it as carbon. Now, it doesn't mean you can't break it down further than that. You certainly can. What we're seeing here is a list of the 3 kind of big subatomic, meaning smaller than an atom, subatomic particles. Atoms are made up of protons and neutrons and electrons. But if I were to reach into that big box of carbon and pull out a proton or an electron, you could not tell me, oh, that's from an atom of carbon. It really is the combination of these subatomic particles that make up an atom that allow you to identify it as whatever type of element it is. Now, obviously, I wrote protons in red here to draw your attention to it. Quite honestly, it is the number of protons that dictate what kind of atom you're dealing with. For example, we're going to talk about carbon a lot today and Wednesday. In every atom of carbon, you will always find 6 protons. And anytime you see any atom with 6 protons, you automatically know that's a carbon atom. I did not write neutrons and electrons in red because, as we will learn shortly, the number of neutrons for atoms of a given element can actually change, and the number of electrons for atoms of a given element can sometimes change. So you have some options here, you have some options here. Number of protons is really what tells you what kind of atom you're dealing with. So that's a big one that we'll always kind of pay attention to. And there's a term in chemistry that we'll see in just a minute that helps us keep tabs on that exact number. Before we learn that term, what I want to do is show you guys a picture of an atom. We're going to focus on this picture. This is called a Bohr model or an electron model. It's basically giving us a way to visualize the parts of an atom and like where they're located, what an atom would look like. Obviously, atoms are super, super, super tiny. You can't actually see it, but this is without some interesting technology, but this is the way that we can represent it for ourselves. Okay. Down below here, we've got a table with all the big important properties of these 3 subatomic particles. So this one slide has a lot of information. Again, if you've taken chemistry before, this is probably stuff you already know. But if you haven't, or if it's been a while, this is kind of like where to start. You got to get these facts down. I'm going to start with the protons, which again are kind of maybe the defining subatomic particle, because based on how many there are in an atom, we know what kind of atom we've got. Protons are positive. They have a positive charge. That's what that +1 means. And they're located right in the middle of an atom in this big cluster in the middle called the nucleus. They're there along with the neutrons, which are neutral, hence the name neutron. So you've got positive protons and neutral neutrons all clumped together in the middle in this nucleus. Our third subatomic particle, the electrons, they have a negative charge, and they are not in the nucleus. Instead, they're in what are called electron shells, or orbitals, if you want to use that term. And they are moving really, really fast. If the nucleus is like right in the middle, picture the electrons like zooming around and around and around. And so the first thing I want to point out is like, why do they not just like fly away from the atom? If they're zooming so fast, why don't they just fly right away? It's the charge differential, right? What do we know about opposites? They attract, right? So we've got a positive nucleus because of the protons, and those electrons are bordering at every second of like flying right away. But when they get a certain distance, that charge differential, right, positive, negative, opposites attract, keeps them associated with the atom. They're moving, moving, moving really, really fast. Because they're moving so fast, it's actually very, very difficult to pinpoint the exact location of an electron at any given moment in time. That's where this version of the figure comes into play. Instead of seeing the electron shell written as this black line that we're picturing the electron moving on, this is closer to reality, because what these little dots are basically giving you is almost like a probability map. Like if you could freeze frame the electron at any given moment, where are we most likely to catch it? So yeah, the line is darkest in the middle, kind of right where that line would superimpose, but every so often when it's circling, the electron will swing in a little bit closer or it will swing out a little bit further. That's a general sense of where it's going to be found, usually somewhere right in the middle of that kind of little map there. We typically simplify and just draw it in this way to help us kind of streamline it a bit. The last property I'm going to go over here that I haven't mentioned yet, atomic mass unit. Remember, atoms make up matter, which means something has mass. So these are the parts that everything is made of. They have a mass associated with them as well. Right, we just talked metric system in lab last week. You guys know that a gram is the basic unit of measure for mass or weight, and yet this is not a gram, right? What is this AMU? This stands for atomic mass unit. You could look up if you wanted to. You will never have to. I couldn't even tell you the number off the top of my head. You could look up how much one atomic mass unit is in terms of grams. It's a really, really little number. Essentially, imagine if I were to tell you to take one proton and set it on that balance in lab, just to see how much one proton would weigh. It would be a ridiculously small number because protons are very, very small. So instead of dealing with a super little number like 0.0000 whatever grams, scientists have just set the weight of one proton equal to one atomic mass unit. because the number 1 is a lot easier for us to work with than 0.000 whatever. Protons and neutrons are around the same size, so they both contribute 1 AMU to the overall mass or weight of the atom. You can see electrons here written as 0. Do you think electrons actually have 0 mass? No, they do weigh something. But even compared to protons and neutrons, which are already small, compared to the mass contributed by those, electrons are so much smaller that we can virtually ignore the contribution they make to the mass of the atom. So to make our lives easier, we essentially just set this at 0. We don't have to worry about it because they're so tiny. So if you wanted to know how much an atom overall weighed or how much mass it had, you would essentially count up how many protons it's got, count up how many neutrons it's got, and that would give you the overall view of things. Okay? I'll ask you that of this atom in just a couple minutes, but I want to first kind of go back to what I said a minute ago about how the number of protons is so important, so we assign a certain vocab term to describing how many an atom has. Okay? If we wanted to describe or picture any kind of atom, what we could do is draw this kind of a picture. This is one of the simplest atoms that we're looking at. Some atoms have many more protons, many more neutrons, many more electrons. So it would, if I'm being honest, kind of be really annoying if we had to draw a picture like this every time. Luckily, we don't. There's an abbreviation system. That's what atomic symbols are all about. So with atomic symbols, what we're going to see is kind of a shorthand way of describing the name of an element, and also a shorthand way of capturing that important information that we went over on the last slide, namely how many protons does this atom have and how much mass does it have, which is protons plus neutrons. So with this atomic symbol, we're going to get away without having to draw the whole picture out, that we can capture all of that important information about any given atom. Okay, the first part of this abbreviation is going to be 1 or 2 letters. Sometimes, like for carbon, it's a simple like, oh, C for carbon, right? Same thing for hydrogen. It's just the first letter the name of the element starts with. When it's 1 letter, like the C for carbon or the H for hydrogen, it's always capitalized. Other elements have 2 letters in their abbreviation. If there are 2, bless you, the first one is capitalized, the second one must be lowercase. Now, you can tell that sodium, like where the heck is the Na from? Not all of these match up in the same way that you would predict. Some of them are a little bit trickier. The good news is I'm never going to ask you, like, what's the abbreviation for mercury? Like that, right? You might have to memorize the periodic table for chemistry. You do not have to memorize it for this class. The ones that we see a lot, you'll probably just memorize because we see them so much, like C for carbon, H for hydrogen. Just really a handful of them that we're going to see so often, and the rest we'll always define when we need them. What I mentioned on the last slide is that each atom, because of the numbers of protons and neutrons it has, each atom has a specific mass or weight, if you want to think about it that way, associated with it. And that is one of the things we keep track of. Really, there are 2 important things we want to capture with this abbreviation system. The first is the atomic number. This is the number of protons that an atom has. Remember, the atomic number is so important because this is really what tells us what kind of atom we're dealing with. Right? Remember I said a few minutes ago that every carbon atom everywhere in like the universe has 6 protons. Its atomic number would be 6. So as soon as you find any kind of atom anywhere that has 6 protons, you automatically know it must be carbon. But you also know that thinking about the mass or weight of an atom, we have to think about more than just the protons. Because you know protons and neutrons are about the same size. They both have an atomic mass of 1 AMU. So if we want to think about the mass or weights of the atom overall, we have to include both the protons and the neutrons. Now this number, protons plus neutrons, 2 different names you can call this, atomic mass or mass number. I don't know why, I always had a hard time keeping these two straight. I just always got them mixed up in my head. So here's how I ultimately got it right. Whichever term you use here, they both say the word mass in them. So I think mass, weight, what contributes to the weight? Oh yeah, it's protons plus neutrons, right? So if you see mass as part of it, you know it's got to be protons plus neutrons. If it's just the number, it's the number of protons. Okay. So we know we're going to have an abbreviation for the name of the element, and we're going to try to capture both of these numbers in some way. So here's one of these atomic symbols, right? So this one is for carbon, that's what the C is for. The bottom number is the atomic number. So let's just make sure we've got it. The atomic number tells us the number of protons, good. And then to get the mass number or atomic mass, it's protons plus neutrons, good. So hopefully we all realize that even though the number of neutrons is not written somewhere here specifically, could we figure it out? Yeah, right? This is protons plus neutrons. This is just protons. So if you do a subtraction, you'll figure out how many neutrons are in this atom. So how many neutrons does this carbon have? 6, right? Because 12 minus 6 is 6. Good. So with this kind of summary or shorthand way of talking about carbon, we've got the number of protons and the number of neutrons. Well, you have to kind of infer here. This is written later, but I'll say it now. Typically, unless you're told otherwise, typically atoms have the same number of protons and electrons. Same number of little pluses and little minuses that would cancel each other out. Most atoms are overall neutral. So knowing that carbon has 6 protons, how many electrons would you expect it has? Also 6. Okay, so again, usually atoms have same number of protons and electrons, positive and negative. What we're going to do next is I'm going to show you that picture we started off with a few minutes ago. And I want you to look at this picture and see if you can tell me the atomic number for that atom. What do we think? Atomic number is the number of protons. How many protons are in that atom? 2. So the atomic number is 2. This is where if you had a periodic table, you could look up, okay, what kind of atom has 2 protons? Helium.
Good luck with today's lesson.